Good afternoon, and welcome to the Center Stage Seminar Series event. My name is Tiffany Willoughby, Education Programs and Outreach Manager in the Office of Research at the University of Texas at Dallas. I will be your moderator. Joining me today is Dr. Michael Rugg. Dr. Rugg is a researcher in cognitive neuroscience and human memory and serves as the director for the Center for Vital Longevity. His research aims to improve understanding the basic mechanisms of memory formation and retrieval, and how aging, injury, and disease impact memory. Please join me in welcoming Dr. Rugg as he takes center stage in the spotlight. Dr. Rugg, welcome.
Thank you very much. So, welcome everybody, and thank you very much for listening to this presentation, and thanks to the Office of Research for inviting me to be part of this series. What I'm going to do today is to give an overview of the Centre for Vital Longevity. I'm going to spend a little bit of time talking about the centre itself and our main sort of mission. I'm going to spend a little bit of time talking about why what we do is important both scientifically and from a translational point of view. And then I'm going to give an overview of the research programs that are running in the centre. And I'm going to give a little snapshot of the kind of work that's done in each of our 7 labs, which are directed by our 7 different principal investigators. So without further ado, essentially the unifying theme of the CVL is the idea of trying to do research that will improve or lead to cognitive health for life. So our research is focused on a bunch of different topics, but the underlying themes that cut across all of our laboratories are trying to understand how age-related changes in the brain's structure and function lead to corresponding changes in cognition. So as I'll show in a little bit more detail later, even in people who are growing old without disease and growing old in what one might call a healthy way, various kinds of cognitive abilities decline as we grow older. We'd like to know why that is the case, what is going on in the brain that underpins this age-related cognitive decline, and obviously, ultimately, is there anything we can do to ameliorate it? As part of that, we'd like to understand why, even in the absence of disease, some people demonstrate less age-related cognitive decline than others. Many of the figures that one sees, and in fact, one of the ones I'll show today, are really kind of averages. They are what happens when you take a large group of people and average their scores, for example, as a function of age. But when one looks in detail, there are striking individual differences in the rate with which different people decline in their cognitive abilities as they grow older. Obviously, if we could understand what the basis of these individual differences are, then we would have some insight perhaps into how those people who don't age as successfully as others could be helped to age successfully. More translationally, we're also interested in trying to identify brain markers that will predict who's most likely to maintain cognitive health and who's most likely to be at risk of falling victim to Alzheimer's disease and other causes of neurodegenerative disease that come on with age. This is critically important. By far and away, the biggest cause of pathological aging and pathological cognitive decline in later life is Alzheimer's disease, and it's been estimated, in fact, the estimates vary, but some estimates suggest that by the age of 85 to 90, as many as 25% of individuals are showing some signs of Alzheimer's or related dementias. Critically, we would like to be able to identify who is at risk of Alzheimer's as early in the lifespan as possible, so that interventions can take place before, if you like, the damage has been done. For the foreseeable future, at least, I think it's unlikely that we're going to be able to replace dead neurons. One of the features of Alzheimer's disease is neuronal death, and if we want to help people who are falling victim to the disease, we need to do so as early as possible, so as to prevent that kind of damage from occurring. And finally, we have research programs in the centre that are targeted at looking for interventions that might slow or even reverse age-related cognitive decline. And I'll give one of my colleagues is just undertaking a large clinical trial right now, which is aimed at this kind of an approach. And I'll talk a little bit about that as we go on. So just a little bit about the centre itself. The CVL was founded by my friend and colleague, Dr. Denise Park, in 2010. In fact, we celebrate our 10th anniversary very soon. And Dr. Park began founded the centre as the founding director. I joined the centre around about 2011 as co-director. And as things stand at the moment, I'm the director of the centre and Dr. Park is the director of research. And between us, we do our best to guide the centre through these increasingly interesting times. We are housed in a private building, shown here, Viceroy Drive. I'll show where it is in just a moment. We occupy the top floor and most of the 7th floor of this building, and the centre as a whole, we house about 70 researchers and support staff. We have about 30,000 square feet of lab and office space in this building itself. The building houses around about 7 research labs. We're actually located here. This is UT Southwestern down here in Harryhausen Boulevard. We're located here. We look out over the lush lawns of the Rugg Hollow Country Club, and we are about, I guess, a mile and a bit away from UT Southwestern Centre for Brain Health. And this is quite important for us. Much of our research involves neuroimaging, and we make heavy use of the neuroimaging facilities, both at UT Southwestern and also going forward, we will be making use of the new imaging facilities that have come online at the Centre for Brain Health as well. In terms of our funding, the majority of the funding for the centre comes from federal research grants, primarily grants awarded by the National Institute on Aging. At the moment, 6 of our 7 labs have support from the NIH, from the NIA, and currently we're holding approximately $20 million worth of federal grants. And the grants are really the fuel that drives most of the research that I'm going to be talking about. So why is cognitive aging important? I'm sure many people are familiar with figures like this and familiar with the issue, but this, I think, puts it into fairly stark relief. We're looking here at a population pyramid for the United States, circa 2012, where we're looking at the numbers of people in the US as a function of age. And we see this pyramid after about age 60, the proportions of individuals declines fairly dramatically. And as of 2012, around about 29 million people in the US were aged over 70 years. About 9% of the population. By 2035, in the US, about 53 million people, almost twice as many people, will be aged over 70 years. And people aged over 70 years will be 15% of the population. So this is a classic example of what one might call an aging society. The US is by no means the society that is aging most rapidly or most acutely. For example, Germany and Japan have much more striking statistics than we do. And this phenomenon is very much a global phenomenon. It's equally evident in developing countries as it is in industrialized countries. So anything that is varying with or correlating with increasing age is obviously becoming more and more of an issue going forward. Why is this important if you're interested in the mind and if you're interested in cognition in the brain? One little example is shown here, and this is a celebrated figure. Could be celebrated for its sparseness as much as anything, but it's one of the best estimates using longitudinal data of what happens to memory function as we grow older. This comes from a very large and long-lasting longitudinal study that's been conducted in Sweden over many years now. And what we're looking here at is the change in memory score as a function of age. And what you can see is that as people get older, around about the age of 60 onwards, memory function begins to decline, and this decline is positively accelerating. So the bottom line is that even people who are aging healthfully, without any evident signs of disease, as they grow older, for example, in this case memory, but it's equally true of many other cognitive abilities, is declining in an accelerated fashion. As we have a larger and larger cohort of individuals in our population over the age of 60 and increasingly in 70s and 80s, then clearly we're having a large number of people who are beginning to experience some degree of cognitive decline. And it doesn't take much of a stroke of imagination to think it would be a good thing if we could understand that and do something to slow or ameliorate it. Equally important, if not more important, are the effects of age and on Alzheimer's disease, which I'll come to in a moment. But first, I should just show you this figure. This is a figure, again, a longitudinal study. Every one of these lines is an individual person. What we're looking at here actually is a study of people who suffer from Alzheimer's disease. It's a very unusual form of Alzheimer's disease. It's called familial Alzheimer's disease. And this is very rare, but it's a form of Alzheimer's disease which is caused by a mutation in one of 3 different genes. If you carry this mutation, you are essentially certain to develop Alzheimer's disease, and you do so at a relatively young age. What the researchers have done here is to follow a large number of people who carry one of these mutations and to measure their memory at regular intervals and to track the moment in time at which memory begins to decline on average relative to the moment in time at which individuals on average are defined as being clinically demented. That is, they're no longer able to look after themselves and they would fulfill the criteria for dementia. And the critical point here is that you can see the decline in memory function onsets more than 10 years before the emergence of what will be a diagnosis of clinical dementia. In other words, the changes in the brain that are eventually going to lead to debilitating cognitive decline are beginning some 10, 12 years, as we just simply measure it relatively crudely, for this moment of clinical crisis. What this tells us is that if we want to do something about helping people who are likely to fall victim of Alzheimer's or other similar diseases, we want to be intervening early. There's no point waiting till symptoms are very clear. We need to be going back in time and intervening with people prior, preferably, to when any of these cognitive symptoms even begin to emerge. So this puts a premium on efforts to identify as early as possible in the lifespan those people who are going to fall victim to Alzheimer's. I emphasize that this is an unusual population. Most people who develop Alzheimer's disease do not do so in middle age. And in fact, the next figure here demonstrates what the prevalence of Alzheimer's disease is as a function of age, generally speaking. And for most people in what's called sporadic Alzheimer's disease, the likelihood of developing the disease goes up as a function of age. And in fact, the biggest single risk factor for Alzheimer's disease is age, is increasing age. And the probability of developing Alzheimer's goes up essentially almost exponentially as a function of increasing age after about the age of 70. Critically, if we could delay the onset of Alzheimer's disease just by 5 years, we would roughly halve the number of cases in the population. And the reason for that is that Alzheimer's is a disease of old age. And so if we could delay the onset by 5 years, many people would pass away before they developed the disease. So critically, when we're thinking about ways of ameliorating the burden of Alzheimer's disease on society, we need not just to be thinking about interventions that are what one might call a cure, if such a thing could exist. We need to be thinking about interventions that slow the progression of the disease. If we could do that, it would have an enormous impact. And we'd like those interventions, as I said, to be ones that can be administered to people as early in their lives as possible to give them the maximum time of cognitive health. And so, although the onset of Alzheimer's disease clinically is coming on around about the age of 70, 75 with significance, we want to be looking back here to find those people who are going to be these people later on, so that we can shift this curve as far backwards in time as possible. So in the centre, all of our research groups use a variety of different methods to different degrees. I'll just spend a little bit of time talking about what those methods are. All of us use traditional methods of experimental and cognitive psychology to try to understand something about the cognitive processes that we're studying. But we have a variety of different methods that are used to image or to manipulate the brain itself. The most important of these methods, and the one that's used by every one of our research centres, is magnetic resonance imaging, or fMRI, magnetic resonance imaging, MRI. This is a stock photo of a Siemens MRI scanner. Once you've seen one of these, as people will know, you've seen them all. But this is an MR scanner, and we can use this in two principal ways. One of these is to obtain images of the brain structure. So, for example, this is a picture or an image of the brain, so-called T1 weighted image, that emphasizes the grey matter of the brain and can be used to measure the thickness and the volume of different grey matter structures. This is a different kind of structural brain image that allows us to look at the white matter connections between brain regions. So what this is doing is imaging the white matter that connects different brain regions. This is the corpus callosum, for example, that connects the two hemispheres. And these techniques allow us to quantitate the integrity and of these white matter connections, and to ask questions about individual differences in the integrity of these connections, or how these connections vary as a function of age. In addition, MRI can be used to obtain images not of brain structure, but brain function. So what we're looking at here is a structural image of the brain superimposed on which are regions which increase their activity, in this case, when someone succeeds in remembering information compared to when they fail to remember information. So this is a functional MRI image. This is obtained by having people lying in the scanner doing experimental tasks. And what we're seeing here is an example of the circuit that is activated when people succeed in remembering prior events. We see the hippocampus, which many people will be familiar with as an area that's important for memory, along with medial prefrontal cortex and the posterior cingulate. And these sorts of functional images we use to a great extent, particularly in my lab, but other labs also, to try and understand something about the organization of memory and other cognitive processes in the brain and how they change with age. In addition, functional MRI can be used in a somewhat more sophisticated way, arguably. We can, not me personally, but Dr. Wig in particular in the lab has developed or has pioneered methods that allow us to take a large number of brain regions And by looking at how activity spontaneously goes up and down in the brain while people are lying in an MRI scanner, to make inferences about how strongly those different brain regions are interconnected. And then by using analytic methods like graph theory, it is possible to generate these sorts of figures that tell us about which of these brain regions appear to be clustered together to form functional networks. And what we can see here is a series of different functional networks which are strongly interconnected amongst themselves. The nodes of these networks are strongly interconnected and rather sparsely interconnected with other networks. And there are a whole variety of methods or measures that can be used to characterize these networks. And as I'll show later, it turns out that this is a very interesting way of analyzing the brain if one is interested in changes in the in brain activity over the lifespan. Another important method that's used in the centre is positron emission tomography, or PET. This involves introducing a radio tracer into the bloodstream of participants, and the radio tracer can then be used to identify and to quantify different sorts of features of the brain. And what's particularly important for a couple of the groups in our centre are methods that allow the quantification of the build-up of two critical proteins in the brain that are involved in Alzheimer's disease. So here we're seeing a PET image where the redness of the image is proportional to the amount of beta amyloid that is accumulated in the brain. Beta amyloid is one of the proteins which accumulates in pathological quantities in people with Alzheimer's disease. This is a participant with Alzheimer's disease, and you can see that the brain is pretty well full of amyloid. Tau is another pathological protein which is found in Alzheimer's disease. And more recently than the development of ligands for amyloid, there are now ligands that bind to tau, so it is possible to quantitate and localize the buildup of both beta amyloid and tau in the brains of living people. And this has turned out to be something of a game changer for the study of the development of Alzheimer's disease. And I'll give an example of that in just a moment. Two other methods that we use in the centre. One is EEG. We can record the electrical activity of the brain from individuals. And we have a research program which examines EEG-based measures of brain activity as a function of age, and particularly in the context of memory experiments. And finally, we have a lab which allows us to carry out a method called transcranial magnetic stimulation. And this involves the targeting brief magnetic pulses to the brain. And the lab allows us to identify a specific region of the brain where we want to target this magnetic stimulation to. The magnetic stimulation can be used either to suppress the function of a specific brain region or to enhance the function of the brain region, depending on the parameters of the pulse sequence. And this allows us to actually gain control, in a sense, over brain function, and to allow us to address questions such as whether disturbance of a particular region of the brain has the predicted effect on a particular kind of cognitive task. So these are the sorts of methods that we have available to us in the centre. And before I go on to talk about the research labs, Tiffany asked me if I would pause just in case there are any questions at this point.
Thank you, Dr. Rugg. I appreciate that. There are a few questions in the queue. The first question that I have for you is, has COVID-19 affected research goals and focuses for the Center for Vital Longevity? And if so, in what ways?
Well, thank you for that question. Yes, it's affected us pretty dramatically. We have been working remotely, as has the rest of the university, since I think the third week of March. And as things stand still at the moment, we have a suspension of human subjects research. So as things stand, we have not been collecting data since the third week of March in almost any capacity. So that's had quite an impact on our data collection programs in all of our labs. And we have been focused, all of the labs have been largely focused on the development of new studies and the analysis of existing data. You can imagine that we are very anxious to get back to human subjects data collection. We expect that's going to be a fairly protracted process, and it's one that we hope that we can get underway very soon. But yes, we have been pretty dramatically impacted.
Thank you, Dr. Rugg. Next question, do environmental factors play a role in the development of Alzheimer's disease, and do you have suggestions for disease prevention?
There is controversy about the role that environmental factors may play in the development of Alzheimer's disease. It's possible that there are some kinds of environmental pollutants that may play a role. In general, though, the thought is that these factors, they may be statistically significant, but they're probably not the major players in what increases your risk of Alzheimer's. In terms of what can you do, there is a joke, which is not a very good joke, but it's choose your parents wisely. So, but putting that to one side, the best advice that I could give is to keep yourself in good shape and to follow the general advice for staying fit and keeping your cerebrovascular system fit. And just remember the adage that what is good for the heart is good for the brain. It's possible also that staying socially engaged and cognitively engaged as we grow older may also play a beneficial role, but the jury is somewhat out on that at the moment. So it's difficult to give a definitive answer there. But basically, the advice I would give is to stay as fit and as engaged as you can.
Thank you. Next question, what can be done to delay Alzheimer's or other cognitive diseases?
So again, it's a great question. And the short answer to that right now is we don't know. We are, along with many other research centres in the country and in the world, we would very anxious to answer that question. At the moment, though, no one could put their hand on their heart and say they know how to intervene to delay the onset of Alzheimer's.
Thank you, Dr. Rugg. We do have a few additional questions in the queue. Is urbanness and fast pace of life a major cause of Alzheimer's, and is medicine the better cure or behavioral changes?
So at the moment, the different factors that drive Alzheimer's or cognitive decline, the environmental and lifestyle factors, that is a very, very active area of research. And at the moment, it would be very difficult to say whether an urban, fast-paced lifestyle, you could say, yes, that's going to be good for you because you're very engaged. It might be bad for you because it's very stressful. And the kind of research that needs to be done to understand those issues is still, I wouldn't say it's in its early stages, but it certainly hasn't come to any definitive conclusions yet. I will describe a project being undertaken by my colleague, Dr. Wig, later on in the talk, which kind of gets at exactly this kind of a question. In terms of the medicine versus lifestyle, right now, there are no interventions that we know of that ameliorate your risk of Alzheimer's disease, in the sense that there's no magic bullet here, there's no magic wand. I could not say that there is any medicine that you could take that will reduce your risk. I couldn't say that there is any specific lifestyle adjustment you could make beyond the advice to stay fit and healthy and to look after your cardiovascular system and hence look after your brain.
Thank you. how does a person get involved in one of your studies?
So you can go to our website, and if you click, I believe there is a tab that says participate. And if you click on that, you will get the opportunity to go to one of any of our research groups or our research groups that are currently recruiting participants. And you can contact a research group, and if you're interested potentially in volunteering, contact the group and they will get back in touch with you and discuss the opportunities for participating.
Thank you, Dr. Rugg, and I will post that website link in the queue.
Thank you.
So that anyone who is interested can visit it. I do have a few more questions in the queue for you. You have a very interesting topic to discuss. How, if at all, do labs in CVL use computational techniques to analyze data that has been collected? And what opportunities for undergraduate researchers exist to do research in CVL labs remotely? And I believe you've answered the second question.
Yeah, so all of our labs make heavy use of computer-assisted analysis, obviously, and some of this analysis is quite advanced, quite cutting edge. To give just one example, which is not meant to overshadow others, but I just know about it, we use machine learning methods in my laboratory, for example, as one of the methods that we use for analyzing our data. Anyone who's interested in contributing or participating as a researcher, undergrad researcher in our labs, I would say go to the website, look at the labs and get in touch with the PIs to see what the opportunities are.
Thank you, Dr. Rugg. There are a few more questions in the queue. Let's go ahead and take care of the next three, and then we will carry on with your presentation.
Okay.
All right. Is mindfulness meditation one of the intervention methods suggested to use with Alzheimer's, and has there been research in this area?
So this is my ignorance. I do not know how much research has been done examining the impact of mindfulness training in dementia or Alzheimer's. This is just something I don't know. More generally, I know there have been a lot of efforts to investigate the impact of mindfulness in cognitive well-being more generally. And my understanding is that those results have been pretty encouraging. But I can't speak specifically to the impact of mindfulness training on dementia and Alzheimer's.
Dr. Rugg, do you forecast a cure for Alzheimer's or simply cognitive health improvement?
So I think cure is a strong word to use. That would be ideal. If by a cure you mean we could give someone an intervention that meant that if they had the disease, they now don't have the disease anymore. I think that's a hard thing to imagine. As I said, at the moment, if you develop Alzheimer's disease and you're developing clinical dementia, that means that you have lost quite a lot of brain tissue. And at the moment, we don't know of any way of regenerating neurons. In the adult human brain, neurons essentially, once they die, they're gone. They do not get replaced. So the effort is not so much on a cure as on prevention. And as I was saying, in an ideal world, we would want an intervention that simply halted the progression of the disease very early in someone's life. If we could slow the progression, though, significantly, that would have a huge impact, as I was saying. And at the moment, it's really all hands on deck. There are a lot of efforts going into clinical trials with different kinds of drug candidates, but by no means is it the case that behavioral interventions won't also be important. And it may turn out that the ideal intervention is a mixture of a drug along with lifestyle and behavioral interventions also. But that's very much in the future, although it's an area that's being very actively pursued right now.
Thank you very much, and I will pass the gavel back to you to carry on with your presentation. Thank you very much.
Okay, great. Thank you. Thank you for the questions. Right, so let me just say a little bit about our individual research labs and the kind of work that's going on in them. So as I said, we have 7 research labs. Each lab is led by an individual scientist, an individual principal investigator. And at the moment, as I said, 6 of these labs are receiving NIA funding. So these are our investigators in alphabetical order. Dr. Chandra Malika Basak, Dr. Kristen Kennedy, Dr. Denise Park, who we met already, Dr. Karen Rugg, yours truly, Dr. Kendra Seaman, and Dr. Gargan Wig. All of us have been with the centre since at least 2013, apart from Kendra, who joined us just last August 2019. And we were delighted to welcome her to the centre. All of us are professors in the School of Behavioural and Brain Sciences, and we have our academic appointments in BBS, and we also teach in BBS, and our graduate students are enrolled in BBS graduate programs. So we have a very close relationship, symbiotic almost, with the School of Behavioural and Brain Sciences. Denise and myself are full professors. Gargan, Karen, Kristen and Chandra Malika are associate professors, and Kendra, who's just arrived, is an assistant professor. And we all have our research groups who are mixtures of research assistants, postdocs, graduate students, other sorts of trainees. And this is what produces an incredibly vibrant scientific atmosphere and culture at the centre. All of these folks are in the centre along with the PIs, and this produces a very, very collaborative and collegial atmosphere for people to work within. It's a pleasure to work with that. I wish I was working there now. Anyway, so in terms of what we do, let me just go through each of the labs. And all I'm going to do here is give a very brief snapshot of the kind of research that goes on. In no way am I describing comprehensively the research programs of each of these labs, but I just want to give just a little vignette of the kind of work that's going on, so you get some sense of the variety of the work and the sorts of questions that we're addressing. So in the case of Dr. Basak, the question here is, can strategy-based video games improve neurocognitive functions in older people? So Dr. Basak has worked for much of her career on looking at cognitive interventions that have a lasting improvement or produce a lasting improvement in cognitive abilities in people. And she has put a lot of work into different kinds of video game interventions. And right now she is embarking on a major clinical trial funded by NIA, which is aimed at investigating on a large scale whether or not these sorts of interventions are beneficial. So in her study, the guts of this are to have a large sample of older adults who are assigned to one of 3 different kinds of training regimes. They will go through training for a considerable amount of time, and they are tested on their cognitive abilities both before and after this training period, and then sometime later. And also MRI data is acquired before and after the training. And the question is whether or not these interventions are beneficial in terms of cognitive ability, and whether there are any correlated changes in the brain that go along with that. So the training system, the training program is what's called adaptive or closed loop. This means that the difficulty of the training that subjects are undergoing is tailored to how well they are doing. So it's continually being adjusted to help them maintain a stable level of performance and hopefully improve their performance. The outcome measures, as I mentioned, include neuroimaging measures, and critically, the question is whether or not age differences in cognitive function, there is a younger group here, a comparison group, whether these age differences in cognitive function can be ameliorated with this video game intervention. Dr. Kristen Kennedy, again, for much of her career, has been focused on structural and functional changes that occur in the brain with aging. And for quite a while now, she has been involved in a fairly large-scale longitudinal study of this. And just by way of an example of the kind of work that she's been doing, this is a recent publication from her lab, looking at this method of what's called tractography. I've mentioned it briefly previously. This is using an imaging method. I seem to be capable of controlling my mouse. Forgive me. This is an imaging method that allows for the identification of white matter tracks. In this case, white matter tracks that are originating in the striatum of the brain and terminating in the frontal cortex. And the question is whether or not measures of the integrity of these white matter tracts tell us anything about age or tell us anything about age-related changes in cognitive function. And what the investigators showed in this study, this is only a very small part of the results, and if you go to the whole paper you'll find a great deal more, is that two measures of the integrity, the functional integrity of white matter, a measure called fractional anisotropy and another one called mean diffusivity, change with age. And in particular, FA goes down with age, which is bad news. Lower FA means a less well-functioning white matter system. And mean diffusivity goes up with age. And that's also indicative of a decline in the integrity of white matter as one grows older. And these are in these specific white matter tracks. And importantly, it turns out that age moderates the impact that mean diffusivity has on a measure of executive function. Executive function is a broad cognitive process that encapsulates such abilities as the ability to concentrate, the ability to hold things in mind, the ability to focus your actions on a specific goal, to control your behavior and basically keep on track. And what these data show is that for older individuals, the higher your mean diffusivity is, the worse is your executive function. Whereas in middle-aged and younger individuals, the same relationship is not found. So it turns out that it's in older people that this measure of white matter has the most relevance to cognitive function. And it's these sorts of data that allow us to put the puzzles of the jigsaw together to understand what's going on in the brain as we grow older that is impacting our cognitive abilities. Dr. Park has been engaged on one of the longest running such studies, I believe, in the world, called the Dallas Lifespan Brain Study. This study actually started, I think, slightly before she founded the Center for Vital Longevity. And it studied some 400 people from across the adult lifespan, healthy people, who have been followed up repeatedly over what's now a 12-year period with neuroimaging and with various kinds of cognitive testing. And one of the critical questions that she's been focused on is whether or not one can find evidence of early signs of Alzheimer's disease in people who are middle-aged, and whether that evidence of Alzheimer's pathology has any cognitive consequences. And this is just some of her data taken from this paper here. But what she has done in this study is to look at how levels of amyloid in the brain have changed over time. So every one of these little lines corresponds to the same individual person measured at two different time points. And so you can see among these people here, there are individuals who show relatively large increases in amyloid between the 4 or 5 year follow-up periods. And also she has measured cognitive function at both of these time points. So it's possible to look at the change in this case, the change in memory between point 1 and point 2. And what this graph shows is that those people who show the largest increase in amyloid over this 4 to 5 year period are the people who show the largest decline in memory performance. In other words, increasing amyloid levels, which are thought to be indicative of the onset of Alzheimer's pathology, are predictive of decline in memory function. And the most important result from this particular study is shown here. This relationship here, which is seen in all of the individuals in the study, turns out to be also evident just in middle-aged people, people aged between 30 and 59 years old. In other words, people in middle age who are beginning to show small increases in amyloid in their brain are also showing declines in memory function that are greater than those individuals who are not showing an increase in amyloid. And it's these sorts of data that indicate the importance of intervening very early if we want to slow or halt Alzheimer's disease, and that give us some clues into what, if you like, the canaries in the coal mine might be, that we might be looking for if we're looking for individuals who are at particular risk of falling victim to this disorder. Dr. Rugg is interested in risk factors, biological risk factors like hypertension and the like, that underlie age-related changes in brain structure and its function and its cognition. And in the study that I'm showing here, she has used 3 different imaging methods. Beta amyloid again, PET imaging of beta amyloid, an MRI measure that allows the assessment of iron concentration in the brain. It turns out that accumulating iron in the brain is not a good thing. This is something that's only become appreciated relatively recently. And she also used high-resolution functional imaging of the brain to be able to measure particular brain structures. And in particular, she's interested in these regions here, coded in blue, which correspond to what's called the entorhinal cortex. And the reason that this area is important is that it is the area that is targeted in Alzheimer's disease by the tau protein first. So when tau gets going in Alzheimer's disease, it attacks this brain region first. And what she was able to demonstrate is that it's individuals who have both high iron and high amyloid who show the largest shrinkage in entorhinal cortex over time. In other words, there seems to be a complex interaction here, such that there's some kind of synergism between amyloid accumulation and iron concentration that mediates whether or not your entorhinal cortex shrinks as you grow older. And again, this is the kind of data that is essential for us to get an understanding of what is going on in the early stages of this disease and in people who are at risk of it. In my case, my research program for many years has been focused on the relationship between brain activity and memory. And just very briefly, an example of the kind of study that we have been doing recently is shown here. These are fMRI images that show regions of the brain that respond selectively either to the pictures of complex scenes or to the pictures of faces. And what we were interested in, along with many other people who've done similar studies, is whether or not the selectivity, the degree to which, for example, this yellow region responds more vigorously to a scene than a face, or the degree to which these red regions respond more vigorously to a face than a scene, whether the selectivity varies with age. And what we have been able to show, and I've shown a couple of times now, is that in these face-sensitive regions, face-selective regions, younger and older people show very, very similar levels of preferential responding to faces. Whereas in these yellow regions, the so-called parahippocampal cortex, younger people show much more selectivity in terms of responding to scenes than do older people. And this effect here is extremely robust, and we've seen this, as I say, in more than one study. It's very striking. Moreover, if we look at the level of selectivity in this region here, the PPA, and we ask how much more strongly does the PPA respond to a scene than a face, this predicts people's memory function. So we can predict how good people's memory are for knowing about the level of selectivity that we see in this brain region. And interestingly, the predictability doesn't vary with age. So it looks like what's going on in this region in terms of its selectivity has important implications for understanding age-related changes in the brain that in themselves impact memory function. And we have a whole research program that's exploring the implications of these sorts of observations. Dr. Seaman is interested in age-related changes in sensitivity to reward and reinforcement, the kinds of variables that we all work hard for, and in particular in how decision-making may change as a function of age. And just by way of one of her projects here, we have an experiment in which someone, so-called investor, I should say, is playing what's called a trust game with another individual. And in this game, what happens is that if I'm the investor, I get given $10, and I can decide how much of that $10 I give to somebody else. And when I give that amount of money to the other person, that person receives 4 times the amount I gave them. So if I hold on to $5 and give someone $5, they get $20. This person can now decide whether to hold on to all of the money or to give half of it back to me. And your job as an investor is to figure out what the best strategy would be, given how often this person returns half the money to you. And the trick is to have 3 different people who have what one might call 3 different levels of trustworthiness. So this individual almost always gives you half the money back. This one gives you half the money back, maybe 50% of the time, and this person never does. And obviously, if you do this sort of thing as an investor, you should rapidly realize you really shouldn't give this guy all your money, or virtually any of your money, because you're never going to get it back. Whereas you can make a big profit by investing in this person who's going to give you half the money back. And what Kendra has shown is that in both older and younger people, you get the predicted effect in that the more trustworthy the person is, the more money they invest. But older people are much more trusting than younger people. So older people tend to be over-investing in untrustworthy people. And this has obvious implications for age-related differences in financial decision-making and the likelihood that older people are likely to fall victim to financial scams and the like. And understanding the reasons for these sorts of changes with age is Kendra's program. Finally, last but not least, Dr. Gagan Wig is very interested in all sorts of aspects of brain network analysis. And in particular, he has just embarked on a project asking about the aspects of an individual's environment that impact the aging of their brain networks. This comes back to one of the questions, Tiffany, that you were asking. And what was the precursor of this project is this fascinating data that came from Dr. Wig's lab in collaboration with Dr. Park a couple of years ago, where they looked at brain measures as a function of socioeconomic status and as a function of age. So we have here individuals who are of low or of high socioeconomic status, and sorry for this, and who are either young, middle-aged or older. And what the investigators demonstrated was that in middle-aged individuals, people of low socioeconomic status relative to high socioeconomic status have lower brain system segregation. That is, these brain networks that can be identified are less well segregated. They tend to be more mushed together. And moreover, the thickness of the cerebral cortex is also thinner in these individuals than in individuals of high socioeconomic status. This effect is not seen for younger people, and it's not seen for older people. It's seen in middle age, suggesting there's something crucial going on in middle age. And as a consequence of that, Dr. Wig has just embarked on a NIH-funded major project, which is going to follow up these findings in a longitudinal sample, a community-based sample in the DFW area. And that work is going to get going just as soon as we're able to start human subjects data collection again. And with that, I would like to thank you for your attention and thank you for your time. Back to you, Tiffany.
Excellent. Thank you, Dr. Rugg. I do have a few more questions in the queue, and we have just about 2 minutes left with our program to answer them. One of the questions that I had earlier on, and the individual asked me to hold it until the end, is what's the difference between CBH and CVL? Is CVL just a place for studying cognitive diseases, or does the center also provide therapies?
So we are what would be called a research center. We do not offer any therapeutic or clinical interventions at all. We do work with clinicians. So to give an example, I work with neurologists, psychiatrists, and also neurosurgery on different research projects that are being undertaken in my lab. Dr. Park works also with neurologists and also with neuroradiologists. All of us have clinical collaborations, but we do not offer any clinical services or any therapeutic intervention. Our research would be described as basic and pre-translational research, and we are a separate entity from the Center for Brain Health.
Excellent. Thank you for the clarification and for providing a thorough explanation. Another question in the queue is there an enzyme that can be specified to destroy or cut the beta amyloid and or tau proteins that cause Alzheimer's?
I don't know about a specific enzyme. There are in development and developed a range of different drugs that are intended either to clear amyloid from the brain or to clear tau deposits from the brain or to prevent them from assembling in the first place. The most advanced of these research programs is with regard to the amyloid drugs. And there are a number of amyloid drugs that have been developed and that are undergoing clinical trials right now. The idea is that these drugs in one way or another can attach themselves to amyloid and clear it from the brain. As some of you might know, thus far the clinical trials have not been completely encouraging, but just recently there has been more encouraging news from one of these drugs, and the manufacturers of that drug have recently applied to the FDA for a license for its clinical use. But there is a huge amount of work going on trying to develop different kinds of biological agents that might prevent or might clear these pathological proteins from the brain.
Excellent. Thank you, Dr. Rugg, for sharing your time, talent, and resources with the attendees and community at large. For additional information and a comprehensive list of Office of Research events, please visit research.utdallas.edu. Have a nice afternoon.